you very much, Alexandra, for this nice introduction. And thank you also very much for giving me the opportunity to present my work in this seminar. So chemicals are very important in our daily life. So they can be found, for example, in our personal care products, in medication to treat diseases, and they are, for example, also applied in agriculture areas to control pests. And at some point, they all enter our aquatic environment via different routes of contamination. So in agriculture, so for example, in agriculture areas, urban areas, in industrial areas. This means that they pose a potential risk to all organisms that live in this environment, and ultimately also to us humans. But many of these chemicals that can be found in the environment have only crudely been assessed for their toxicity. So this means that classical toxicity tests have been performed in which you expose the organism to a range of different concentrations, and then it is counted how many of the animals are affected. And so from these experiments, we can derive those response relationships, and we can also derive a few values, such as, for example, the LC50 concentration, which is the concentration at which 50% of the animals are affected. But the problem with these tests is that we do not gain much information about the chemical. And for these tests, large numbers of animals are needed. They are not very sensitive and not very efficient, and they neglect subtle effects, which, however, can be very relevant. So toxicologists have realized this quite some time ago, and about a decade ago, there has been a move in the toxicology field towards a more mechanism-based risk assessment. And during this time, the adverse outcome pathway framework was established. And this concept allows to organize toxicological information across different levels of organization, of biological organization, with the aim to causally link molecular initiating events with adverse outcome of regulatory relevance. So this means if we know which, oops, it's really hard to point. So if we know which molecular events lead to an adverse outcome, we can then design in vitro tests or tests to measure such molecular events in vitro, and then can predict whole animal responses without performing the animal experiment. And such mechanistic knowledge is also important for developing potential biomarkers that can, for example, be used in biomonitoring. So this mechanistic knowledge is really important because it lays a critical foundation for predictive approaches in ecotoxicology. So one group of chemicals which has a particularly high potential of eliciting subtle effects, and for which such mechanistic knowledge is largely missing, is the group of neuroactive chemicals. So they pose a yet unpredictable risk to environmental and human health. So these neuroactive chemicals target the central nervous system or also sensory organs, and they can impair the functionality of neurons or can kill neurons. They can also impair the development of the sensory nervous system. And this can have cognitive, behavioral, motor, sensory, and psychological effects. And this can actually ultimately lead to a reduced survival of these organisms. And so these neuroactive chemicals are also very abundant in our environment. And so in a recent monitoring study in European rivers, which grouped the chemicals according to their mode of action, the group of neuroactive chemicals was the largest group. So to better predict the hazard that these neuroactive chemicals pose on aquatic organisms, it is really crucial that we better understand mechanisms of neurotoxicity. And my team and I work towards the closing of this knowledge gap by investigating effects and molecular mechanisms of neuroactive chemicals in the zebrafish model. And the zebrafish model is particularly well suited for this type of studies because it has transparent larval stages, and you can observe organ development from fertilization onwards. And next to other advantages in this model, the zebrafish larvae also show quite a lot of behavioral reactions already early on. And this is nice because then we can use these behavioral reactions as a readout for the functionality of the nervous system. So for example, around 18 hours, spontaneous contractions start, and around 1 day, the larva becomes touch sensitive and reacts with a burst swim. Around 4 days post-fertilization, the swim bladder is inflated and the larvae start to swim more frequently. And they become sensitive to light and acoustic stimuli. And also, if you put the larvae in a current, they start to swim counter current because they can sense the hydrodynamic flow. At 5 days post-fertilization, the visual system is quite well developed, and we can measure visual behavior reactions. And around 6 days post-fertilization, the olfactory system starts to become functional, and the larva can detect quite a large number of odorants. So we are following an approach that we have described in this review here, in which we expose the larvae to neuroactive chemicals and then observe their behavioral phenotype. So in parallel, we then measure changes in neuronal activity or neuronal structures, and as well as in molecular events, with the aim to afterwards causally link these molecular events back to the behavioral phenotype. And to verify such causal links, we can, for example, use CRISPR-Cas9 gene editing tools to modulate the expression of genes that we identified in this analysis here. And then we can try to link this back to the chemically elicited phenotype. So in this seminar, I would like to present you 4 different projects, and I will also show up a few future avenues. So in the first project, we investigated neurotoxic effects of insecticides, and we first looked at attraction and avoidance reactions towards insecticides. And in the other part, we looked at developmental exposure of insecticides. In the second project, we looked at effects of opioids on larval zebrafish behavior, and in the third project, we were interested in the question why some individuals are more sensitive to chemicals than others. And in the last part, I will present you a few computational tools that we generated to predict molecular mechanisms of neurotoxicity. So for our insecticide studies, we selected 8 different insecticides that are present in Swiss waters. And we selected them based on a list of an Eawag study. So we selected 2 organophosphates, thiazinone and dimetoate, sorry, I have to get used to this one. 2 carbamates, metamyl and pyrimicarb, and these all inhibit the enzyme acetylcholine esterase. We also selected 2 neonicotinoids, imidacloprid and thiacloprid, which activate a nicotinic acetylcholine receptor. And we also selected 2 pyridine-based insecticides for which the mode of action is not entirely known. And to visualize these different mode of actions, I'm showing you the neuromuscular synapse. So this is the synapse that connects the nervous system to the muscle. And if an action potential arrives in this motor neuron, the vesicles that are filled with the neurotransmitter are fused with the synaptic membrane, and the acetylcholine is released into the synaptic cleft. Then acetylcholine binds to the postsynaptic receptor, and this depolarizes the muscle and leads to a contraction of the muscle. Then the acetylcholine esterase enzyme hydrolyzes this acetylcholine, which terminates the signal transmission. So if now organophosphates or carbamides are present and inhibit this enzyme, acetylcholine gets accumulated in the synaptic cleft, which leads to overexcitation of the muscle and final paralysis. And if neonicotinoids are present, they bind to the receptor, which also leads to an overexcitation of the muscle, and it also leads to final paralysis. So this cholinergic neurotransmitter is a very ancient neurotransmitter system, and it can be conserved across different species. And so therefore, all chemicals that target this system have a high potential to also affect non-target organisms. So in this first project, we were interested in whether such patches of insecticides could actually change the behavior of fish. So therefore, we designed a chamber in which we can inject the insecticides on one side and can observe the spatial behavior of the larvae. We were then interested whether the larvae or how the larvae can detect these insecticides. And therefore, we looked at whether olfactory deficient larvae have the same responses. We then were further interested in how these insecticides were perceived, and therefore we looked into the neuronal activity. So to do that, we stained a protein called ERG, and ERG is a protein that gets phosphorylated when the neuron was active. So by looking at the P-ERG-T-ERG ratio, so the phosphorylated versus total ERG ratio, this ratio is actually a readout for neuronal activity. And so we stained for these proteins and mapped the signal onto a larval zebrafish brain atlas. So this is how our chamber looked, and this is how it looks like when we inject the insecticide. Oops, this is different. So we measured the mean displacement before and after injection. And the first thing we found is that our positive control, taurochloric acid, which is a bile acid that has been shown to induce aversion before, also triggered aversion in our setup. So what we further found is that imidacloprid and diazinon, so a neonicotinoid and an organophosphate, they also trigger aversion at 10 micromolar of concentration. But for all the other insecticides, we did not find a behavioral change. So then we looked at how the larvae can detect these insecticides, and therefore we inactivated their olfactory system. So we focused on the olfactory system because this system has been shown to be involved in detection of other substances before. So we inactivated their olfactory sensory neurons by exposing the fish to a low cadmium chloride concentration, which disrupts the olfaction but does not change the locomotor behavior. And in these olfaction deficient larvae, the aversive responses that we observed before were abolished. So this means that the larvae can actually smell these two insecticides. And then we wanted to know which brain areas are involved in the behavioral responses, and we then did the P-ERG-T-ERG staining. And so we found actually that about 200 brain regions were differentially active in this insecticide-exposed larvae, and therefore we focused on the top 10 regions. And so we found the most pronounced changes in hypothalamic regions, which is a region that is involved in the stress response. And we also found activity changes in hindbrain regions, which have been shown to be involved in aversion before. So we think that these insecticide-induced responses that we see resemble aversive responses to changes in water chemistry. So then we wanted to see how the larvae are affected when they're exposed to the insecticide throughout their development. So therefore, we exposed the larvae from day 0 to day 6 to these insecticides, which covers the whole development from a fertilized egg to an independently feeding larva. We then measured their behavioral responses, and also we wanted to see whether these are accompanied by structural changes in the nervous system. We then also were interested in whether these effects could recover, and therefore we placed the larvae back into a chemical-free medium for 3 more days. And to measure the locomotor responses, we used the light-dark transition test. Oh, sorry, I don't know, is this working? Okay, this is supposed to be a video. Yeah, I think it's working. So yeah, in this test, the larvae are exposed to changing light conditions. So first, they are exposed to bright light in which they don't move a lot. This you can see now in the video. But if the light switches off, which you can see now, the locomotor activity dramatically increases. And this is supposed to be a light-seeking behavior. And if we plot now the distance moved over time, we can see this increase in the dark periods. So I don't know if you can actually see this, but it should supposed to be shaded here. Okay. So we performed, so we exposed the larvae to these 8 different insecticides that I showed you before, and we used concentrations that did not induce any morphological changes. And so I'm showing just 2 examples here, but what we found is that all cholinergic insecticides decreased the locomotion, and the strongest effects were seen with the metomyl and thiacloprid. And so we also found is that both non-cholinergic insecticides did not affect locomotion. And then we investigated the neuromuscular structures to see whether they're also changed. We first looked at the growth of motor neurons, and what we found is that, so we did not find a lot of changes, but what we found is that metomyl led to an increased number of branches per axon. And so imidacloprid caused the opposite, so it led to a decreased number of branches of these motor neurons. We then looked at the synaptic connection, which is the connection between the neuromuscular synapse that I showed you before. We looked at whether these are properly formed, but there we did not find any changes. We then also looked at the muscle structure, so the third component of the neuromuscular structure. And we assessed this by looking at the birefringence signal. So if in a highly organized muscle, this signal is very bright, but if the muscle is disorganized or impaired, then you can see a decrease of this signal. And we quantified this signal, and what we found is that with thiacloprid and metomyl, the signal was decreased, which means that their muscle structure is impaired. So, and these were also the 2 insecticides that caused the strongest effects in the locomotor activity. And then we were interested in whether larvae can recover from these effects. And so we were actually quite surprised to find that most insecticides fully recovered already 1 day after we put them in a chemical-free medium. And so one chemical, so you actually can see this here, the depurated larvae come back to control levels. So one insecticide, so thiazinone exposed larvae needed 3 days to recover, and pyrimicarb exposed fish did not recover at all during these 3 days of depuration. And we also looked at whether muscles and motor neurons effects regenerated, and we also found a regeneration there. And then we were further interested in whether there is a critical window of exposure during neurodevelopment, and therefore we exposed the larvae during different 24 hours window during their development and tested their locomotor output with an age-matched assay. So I don't want to go into details, but I just show you the most important findings. So the effects were only visible when the insecticides were present. But for earlier exposure windows, the locomotion came back to normal control levels. this means that we did not identify a critical exposure window for locomotion. And so in a continued, in an ongoing project, we are now further interested in the molecular underpinnings of this recovery process. And therefore, we are performing, so I've teamed up with Xenia Gro and the bioanalytics team, and Melissa von Wil, scientific assistant, is also on board. And we are investigating this recovery process by using transcriptomics, proteomics, and metabolomics techniques. And so we have the aim to identify markers that are predictive of transient and persistent neurotoxic effects. So now I'm switching to another group of chemicals, which are the opioids, and I'll like to present you a few interesting findings. So when we expose the fish to fentanyl citrate, which is an opioid that is used as a painkiller and also a recreational drug. So when we expose the fish for 24 hours to this drug, we found that the larva had massively increased swim bladder. So we were quite surprised by this effect, because what's usually reported is that chemicals affect the inflation of the swim bladder. So they normally have a deflated or a smaller swim bladder. And so therefore we were quite interested in this effect. So we followed this up with other chemicals, and we found that also tramadol hydrochloride, which is as well an opioid, causes the same effects, but also carbamazepine, which is an anti-epileptic drug and has a different mode of action, and the insecticide aldicarb, which is acetylcholine esterase inhibitor, they also cause this effect. And so we're still puzzled kind of why these chemicals with this different mode of action, they cause the same effects. So then we were wondering whether this hyperinflated swim bladder would actually influence the position of the larva in the water column. So therefore Joël, a master's student in my lab, he set up a behavior system to record their behavior from the side. And what we found here is that the control larvae, oh, it's really bright actually. I hope you can see it. So the control larvae actually use the whole water column, and you can see this in this quantification here, which is the mean position over the whole recording. But the fentanyl exposed larvae, actually they stick to the surface and they try to go down, but they're actually pulled back up again. So this fentanyl citrate exposure and actually also tramadol hydrochloride, they trigger surfacing behavior. So then we were interested whether this hyperinflated swim bladder is a consequence or a cause of the surfacing behavior. And we addressed this question by investigating which one of these effects occurs first. So then Joël did behavioral recordings from the beginning of the exposure and for 4 hours, and he found that the surfacing behavior was already observable after 20 minutes of exposure. And he did the same for the swim bladder hyperinflation. So he quantified from the beginning of the exposure, and he found that the hyperinflation only occurred after about 3 hours after the exposure. And we also observed that the swim bladder, so the surfacing behavior occurred at lower concentrations than the swim bladder hyperinflation. So we could already observe this at 0.1 micromolar, while the swim bladder hyperinflation only occurred at 1 micromolar. So this means that swim bladder hyperinflation is a consequence of the surfacing behavior. So we are now, of course, very interested in what actually triggers this surfacing behavior, and there we would like to look at further chemicals and other water conditions. We are also interested in whether this effect is reversible, and Joël already looked into this a little bit, and he found that after 1 hour of exposure and 48 hours of depuration, the larva actually still sticks to the surface. And he also observes, I don't know if you saw it, but he observes that it is actually pulled back up quite a bit, and this means that the swim bladder is still hyperinflated. And so we would like to better understand this connection between the sensing of the water condition, the surfacing behavior, and the hyperinflation of the swim bladder. And also we would like to explore to what extent the hyperinflated swim bladder could serve as an easily accessible endpoint to which a change in behavior and an adverse outcome can be linked to. And so in the next project, we were interested in why some individuals are more sensitive to chemicals than others. And so such differences are actually very important to understand because they can tip the scale between succumbing or adapting to a threat. And so they can also, so populations that consist of sensitive individuals actually have a high potential to become extinct in contaminated areas. And so we were also thinking that comparing sensitive versus tolerant individuals might give us indications about the mechanism of action of a chemical. So we followed this procedure here. So we exposed the chemicals, we exposed the larvae to a chemical and then measured their behavioral output. And based on this, we sorted them into sensitive and tolerant individuals. And then we sent them, so we did a transcriptome analysis in order to identify genes that are differentially regulated in these two groups. So, but we had to first do some groundwork because the behavior of unexposed larvae is actually quite variable already. So this is a problem because it makes it very difficult to distinguish between this natural variability and the chemically elicited sensitivity differences. So Jennifer Fitzgerald, a former postdoc in my lab, she set out to search for conditions in which the behavior is very consistent. She also correlated other parameters, such as, for example, the heart rate and the length of the fish and the swim bladder to the locomotor activity. But she did actually not find a lot of correlations. But luckily, she found a condition where the behavior was very consistent. And this is during the dark intervals between 6 and 7 day old larvae. So this was really nice because it allowed us to come up with such a sorting paradigm in which we first measured were able to measure a baseline locomotion, and then we exposed the fish for 24 hours and measured the locomotion again, and we could then calculate the activity difference. And so those larvae that had a small activity difference were sorted into the tolerant groups, and those with a large difference were sorted into the sensitive groups. So, and Jenny also used these two different chemicals, fentanyl citrate and the anti-epileptic drug carbamazepine. And she then sorted the individuals in these three categories. So we also included a middle category, and they actually segregated nicely in the behavior. She then sent them to the RNA, to the transcriptome analysis. And there we actually did not find a lot of differences because we really analyzed the individual larvae. But we were able to identify two interesting candidate genes that were upregulated in the group that were tolerant towards fentanyl citrate. One gene is the G protein coupled receptor, for which there's not much data available for zebrafish, but the human autolog has been shown to be involved in Alzheimer's disease. And the other gene was a brain-specific transcription factor whose expression is regulated by neuronal activity. We are now in the process of confirming this differential expression in qPCR experiments, and the final aim would be to manipulate the expression of these genes by CRISPR-Cas9 tools and to see whether there is a sensitivity shift in these edited larvae. So having now established such a paradigm, we can also start to ask further questions. So for example, fish in the wild, they are probably never only exposed to one stressors, but there is multiple stressors that they're exposed to. For example, changes in temperature or decrease in water quality. And so an interesting question to ask would be how these different stressors would change or would influence the larva's sensitivity to chemicals. And we could now combine these different stressors with chemical exposures, but this would probably lead to a large number of experimental groups, which is almost not manageable. So we could also look at this from a neuroendocrinology point of view, because when the animals is exposed to a stressor, the so-called stress axis is activated. And this axis is a hormonal cascade that leads to the cortisol, to release of cortisol into the bloodstream. And the cortisol from there reaches the different organs to prepare the body better for this stressful situation. And this stress axis is very conserved among species. You can also find it in a 5-day-old zebrafish larvae. So the idea would be if we can manipulate the release of cortisol, we can simulate such a multiple stressor situation. And in the lab where I did my postdoc, we developed a transgenic zebrafish line in which we expressed a photoactivatable enzyme in these steroidogenic interrenal cells. And this would allow us to manipulate the cortisol release just by expressing the larva to blue light. And so my idea would be in the future to investigate how these different stress levels would influence the larva's susceptibility to chemicals. So now we are moving one level further down. So I've shown you before how we did structural analysis and neuronal activity analysis, and now I will show you how which tool we generated to predict molecular mechanisms of neurotoxicity. And therefore, I teamed up with Anje Šupanik, who is a former group leader at UTOX, and a team of computational scientists from Philip Morris and the Fraunhofer Institute. And so we used a method called biological expression language, with which we extracted scientific knowledge from the literature and converted it to a computer-readable language. And Roman Lee, who was a postdoc on this project, he generated two such causal biological networks, which consisted of molecular relationships that he created from scientific literature. And he generated a network for cardiotoxicity and one for neurotoxicity. And these networks also had a second layer, which consisted of a molecular profile for some of the nodes, and which allowed us to score this network with transcriptomics data set, and then to infer from this which nodes were active. And this would allow us to make predictions about molecular mechanisms. I don't want to go into further detail, but if you are interested in these two networks, I invite you to read these two papers here. And so Anje Šupanik and his team were also able to find a method to directly derive adverse outcome pathways from these networks. And we used the neurotoxicity network to develop two adverse outcome pathways for developmental neurotoxicity. So one was that inhibition of the enzyme FIN-A leads to a decreased cell proliferation in retinal ganglion cells, which leads to a decreased eye size, altered visual function, and increased mortality, and this finally leads to a decrease in population trajectory. And the second AOP, there the inactivation of the GSK beta enzyme leads to increased mortality via defects of the developing inner ear. And so our next steps here would be to experimentally verify these predictions and to finally establish a causal link between this molecular initiating event and the observed phenotype. And this would hopefully lead to the further maturation of our putative AOPs, such that they can be finally used in the future for chemical risk assessment. And so I would like to summarize the talk here. So I showed you that insecticides and neuroactive pharmaceuticals have the potential to influence the behavior of fish early life stages. I showed you that imidacloprid and diazinon trigger olfactory-mediated aversive responses and stress reactions. I also showed you that developmental exposure to cholinergic insecticides leads to a reduced locomotion, and a strong reduction can be explained by defective muscle fibers. But that the locomotor effects in larval stages recover relatively quickly, and we could not identify a critical window of exposure. I also showed you that fentanyl citrate and actually also the other opioid, tramadol hydrochloride, leads to uncontrolled surfacing behavior and swim bladder hyperinflation. And so such behavioral changes are in critical life stages that can actually lead to a reduced survival of the animals. And so I also, in the second part, I showed you how we try to understand the molecular basis of sensitivity and tolerance mechanisms. And therefore, we established an experimental sorting pipeline. And we identified 2 candidate genes that are potentially important for the tolerance towards fentanyl citrate. And to understand such differences is important because an altered chemical susceptibility at the individual level can also affect the fitness of the whole population. And in the last part, I briefly showed you that we generated causal biological networks that help to predict molecular mechanisms of neurotoxicity and cardiotoxicity. And we developed also 2 adverse outcome pathways for developmental neurotoxicity. And so these computational tools really facilitate a move towards a mechanism-based risk assessment. And overall, this research contributes to the better understanding of chemical effects on fish, nervous system, and behavior. So I would like to briefly also mention that we have a very exciting project that just started, but I didn't have time to go into detail of this. This is a collaboration between me, Christine Schirmer, Xenia Groh, Bernhard Ruffer, and Jarno Höckmann from the University of Utrecht. And so in this project, we are expanding the tests, the set of in vitro tests to predict for animal-free prediction of toxicity. And we call this the fish in vitro. And we'd also like to facilitate the uptake of these tests by the regulators. And therefore, we collaborate with these 2 social scientists, Bernhard and Jarno. And we also have 2 PhD students on board. And so Jessica Bertoli will actually develop cell-based models for neurotoxicity assessment. And we are also interested in whether we can recapitulate some of the findings that we made in vivo, whether we can recapitulate them in vitro as well. And I would also like to mention that I have a wonderful collaboration going on with Lily Janssen and 2 of her students, in which we investigate the effects of cyanobacterial metabolites on the different organisms. And so therefore, we recently also started to track the behavior of other organisms, so the snails and the caterpillars. And so I also want to mention that if you, so I'm very open to collaboration. So if you see any interesting overlap, please approach me. So, and in the end, so getting tenured at Eawag is not something that you can do alone. And so I would really like to thank the people who contributed significantly to this success. So first and foremost, Sarah Könemann, who was a PhD student in my lab, and she was really the main driver in the insecticides project. And she also established a lot of techniques and assays in my lab. She trained 2 master students, Melissa von Wil and Stephanie Meyer, and Alexander Beetz was involved in data analysis. And then Jennifer Fitzgerald, she was the main driver of the sensitivity mechanism project, and she also initiated the swim bladder project. And I had a master student and a bachelor student, Joël Trummer and Aida Kiani Fard, who both worked on the swim bladder project. And so the computational tools were generated in collaboration with Anje Schupanek and Roman Lee, and a few scientists from Philip Morris and the Fraunhofer Institute, and Aquatox Solutions was also involved there. And of course, this wouldn't also have been possible with a lot of support, with a lot of technical support. So Pascal Bucher is our aquaticum technician who keeps our fish happy every day. And René Schöneberger was performing the chemical analytics. And I would also really like to thank Christine Schirmer for constantly supporting me throughout my tenure track and for being really an excellent mentor. And I would also like to thank the UTOX crowd for creating such a nice and supporting working environment. And now I'm happy to take your question. And thank you for your attention.